Hello, welcome to our explanatory video about the process of gamete formation and non-disjunction. Let's talk about reproduction. Reproduction is a fundamental part of life. It's how living beings pass on their traits, ensuring that life continues from one generation to the next one. But have you ever wondered how this fascinating process actually works in our bodies? Let's take a closer look. Reproduction starts at the cellular level. Have you ever wondered how you inherited your dad's curls or your mom's dimples? Well, it all begins with the process of gamete formation. Inside our bodies, specialized cells called gametes, sperm in males and eggs in females, are created through a process called meiosis. Meiosis is split into two parts. There's meiosis 1 and there's meiosis 2. Let's dive into meiosis 1. It all starts in the prophase. Here the chromosomes condense and become visible as sister chromatids. Homologous chromosomes, one from each parent, pair up. This pairing enables crossing over, where segments of genetic material are exchanged, which creates genetic diversity. Imagine mixing puzzle pieces from two different sets to create a new, unique picture. That's what crossing over does to our genes. The nuclear envelope breaks down and the spindle fibers start forming. Next up is metaphase 1. In metaphase, the chromosomes line up in the cell center randomly. This randomness increases genetic variation and diversity. This is the reasons why siblings look somewhat alike, but also very different. Next comes anaphase. Spindle fibers pull chromosomes apart. Each pair is split, sending one chromosome to opposite sides of the cell. The most important part is the sister chromatids stay together. Finally, in telophase 1, the chromosomes reach the cell's poles, and the nuclear envelope may reform. Then the cell divides in cytokinesis. And there you have it, 2 haploid cells, each with half the original chromosome number. These cells are ready for the next stage, meiosis 2.
Now that we've completed meiosis 1, we have 2 haploid cells, each with half the original chromosome number. But we're not done yet. Meiosis 2 begins. This is where the sister chromatids are separated, creating 4 genetically unique gametes. Let's break it down step by step. First, in prophase 2, the chromosomes in both cells condense again and new spinal fibers form. Next, in metaphase 2, the chromosomes line up at the center of the cell, but this time there are no homologous pairs, just individual chromosomes. Each one is ready to split. In anaphase 2, the spinal fibers pull the sister chromatids apart to opposite poles of the cell. This ensures that each new cell will get a single copy of every chromosome. Finally, during telophase, the nuclear envelopes reform around the separated chromatids and the cells divide in cytokinesis. The result? 4 genetically unique haploid cells, each containing just one set of chromosomes. This will go on to become sperm or egg cells, ready to combine and create new life. But what happens if meiosis doesn't go as planned? What if there's a mistake during this complex process? These mistakes are called non-disjunctions, and they can lead to missing or extra chromosomes in the gametes. When a gamete with an abnormal number of chromosomes combines with a normal one, the resulting zygote will also have the wrong number of chromosomes. This can lead to a variety of conditions, one of which is Turner syndrome. Let's take a closer look. Turner syndrome is a rare condition that exclusively affects females. Normally, females have 2 X chromosomes, but in Turner syndrome, one of the X chromosomes is completely or partially missing. This affects how the body grows and develops. So what causes Turner syndrome? It often starts during meiosis or mitosis. In monosomy X, the X chromosome is completely missing in all cells. This might occur due to an error in the formation of the sperm or egg. In mosaicism, some cells have 45 chromosomes, while others have the typical 46. This can happen after fertilization when cell division goes awry. There are also structural abnormalities of the X chromosome where part of the chromosome is missing. And finally, isochromosome formation, where one X chromosome contains identical arms, losing crucial genetic material. The symptoms of Turner syndrome vary, but often include short height, delayed puberty, a short neck, and infertility. These features may be present from birth or may become apparent during childhood or adolescence. So, what can we take away? Meiosis is essential for creating gametes, ensuring diversity and offspring. It consists of 2 parts, meiosis 1 and 2. Errors in meiosis, like non-disjunction, can lead to abnormal chromosome numbers in gametes. Turner syndrome occurs when one X chromosome is completely or partially missing, often due to non-disjunction. Key causes include monosomy X, mosaicism, and structural abnormalities of the X chromosome. That's it for our video. Thank you so much for watching.